Blackbeard, your trusted channel for exploring the wonders of science and nature. I am Aschalo Asba, and today we are diving into Unit 4 Genetics. In this video, we are going to explore how traits are passed from one generation to the next. We will cover everything from Mendelian inheritance to non-Mendelian patterns, sex determination, pedigree analysis, and genetic disorders. So let's get started. Ever wondered why you might look similar to your family members? You still have your own unique traits. It's all about genetics. Gregor Mendel, the father of genetics, studied garden peas and discovered that traits are passed on from parents to offspring in predictable ways. He introduced us to key terms like genes, alleles, dominant, recessive, genotype, and phenotype. Every individual is a mix of traits inherited from their parents. Mendel found that traits are controlled by pairs of alleles. In heterozygote, one allele is dominant and can mask the presence of a recessive allele. That's why, for example, if you inherit a dominant trait like tallness, you will show the trait even if the other allele is for dwarfism. So your overall genotype and which is genetic makeup and phenotype or your appearance are determined by these interactions. A monohybrid cross focuses on a single trait. Mendel crossed purebred tall pea planters, which is represented by capital letters TT, with purebred dwarf pea planters, which is represented by small TT planters, and found that all around all the offspring the F1 generation were tall, which is represented by capital T letter and small t letter, which shows the dominance of tallness. But when the F1 generation self-pollinated, the F2 generation showed a 3-to-1 ratio. 3 tall plants, that means 3 tall plants for every 1 dwarf plant. We often use a p-unit square to visualize these ratios, with a genotypic ratio of 1:1 and a phenotypic ratio of 3:1. Next, we have the dihybrid cross, where we look at two different traits or characteristics at the same time. For instance, Mendel crossed plants with round yellow seeds with those having wrinkled green seeds. The F1 generation was all round and yellow, but when these F1 plants were self-crossed, the F2 generation showed a ratio of 9:3:1. This demonstrates that traits are inherited independently from one another, which brings us to Mendel's law. Mendel's first law is the law of segregation. It tells us each parent contributes one allele for every trait, and these allele segregate into different gametes. His second law, the law of independent assortment, states that the inheritance of one trait does not affect the inheritance of another. These principles were revolutionary in explaining how traits are passed on. Test crosses are used to determine an unknown genotype. Here is how it works. If you have an organism showing dominant trait or characteristic, it might be either homozygous dominant or heterozygous. By crossing it with an organism that is homozygous recessive, you can look at the offspring. If all the offspring shows the dominant trait, then the unknown parent is likely homozygous dominant. If there is a mix, then it is heterozygous. This method is essential for genetic analysis. Sex determination is another fascinating part of genetics. In human, sex is determined by chromosomes. Females are XX and males are XY. During gamete formation, males produce sperm that carry either on X or Y chromosome, while females produce eggs that all carry on X chromosomes. Then, when fertilization occurs, the combination of these chromosomes determine the sex of the offspring. This process is clearly depicted in figure 4.3. While Mendel's work laid the foundation, not all traits follow his simple dominant and recessive rules. In non-Mendelian inheritance, we see patterns like codominance, incomplete dominance, and multiple alleles. For example, in codominance, both alleles are expressed equally, like in the ABO blood group system. And in complete dominance, the heterozygous phenotype is a blend of both traits, such as pink roses from red and white parents. Let's talk about some special cases. The RH factor is a blood group trait that is independent of the ABO system. People can be RH positive or RH negative, and this becomes very important in blood transfusion and during pregnancy. Additionally, sex-linked inheritance refers to traits that are carried on sex chromosomes. Since males have only one X chromosome, they are more likely to express recessive traits found on it, like hemophilia. Not everything is controlled only by our genes. The environment can also influence how traits are expressed, a phenomenon known as phenocopy. For instance, Himalayan rabbits may change their coat color depending on the temperature, resembling different phenotypes, even though their genetic makeup remains the same. This shows how the environment interacts with our genes to shape our appearance. Since we cannot perform controlled breeding experiments with humans, we use pedigree analysis to study how traits and genetic disorders are passed through families. In a pedigree, squares represent males, circle represents female, and the shading shows who is affected by a trait or disorder. Pedigree charts help us determine whether a trait is dominant, recessive, autosomal, or sex-linked, and they are vital in predicting genetic risks in families. Lastly, let's discuss genetic disorder. These are diseases caused by mutation in our DNA, such as diabetes, cancer, Down syndrome, and cystic fibrosis. Genetic disorders can be classified as autosomal dominant, autosomal recessive, X-linked, or multifactorial. Understanding the patterns of inheritance help us diagnose and manage this condition more effectively. To summarize today's lesson on Mendelian inheritance and beyond, we learned that traits are passed from parents to offspring through genes, as first explained by Gregor Mendel. Monohybrid and dihybrid cross demonstrates how single traits and pairs of traits are inherited. Mendel's law of segregation and independent assortment explain the predictable patterns we see in inheritance. Test crosses help determine unknown genotypes, and sex determination is based on chromosome. Non-Mendelian inheritance, environmental effects, pedigree analysis, and genetic disorders add layer of complexity to how traits are passed on. So this is all about today's lesson. All of this deepens our understanding of why we are the way we are. That's it for today's deep dive into Mendelian and non-Mendelian inheritance. I hope now you have a clear understanding of how traits are passed from and how genetic shapes are who we are. And if you enjoyed this video, please give it a thumbs up, share it with your friends, and subscribe to Black Pill for more engaging science content. And don't forget to leave your questions or thoughts in the comment below. I would love to hear from you. Thanks for watching, and until next time, keep exploring and stay curious. Bye.